Good day everyone. This discussion will be all about your experiment 9, Osmotic Fragility Test. But let's define first your anemia. When we say anemia, this is the decrease in the oxygen carrying capacity of the blood, thus having decrease on the number of RBCs and levels of hemoglobin and hematocrit. Now, your RBC count, your hemoglobin, and your hematocrit levels are used to compute the RBC indices, mean cell volume, your mean cell hemoglobin, and your mean cell hemoglobin concentration, which are usually used for the parameters in determining anemia. Now, there are several factors as to why a patient is having a symptom of anemia. It may be due to genetic or acquired factors, environmental, pathological, or iatrogenic factors. You have two mechanisms as to why a patient can have anemia. It may be due to ineffective erythropoiesis or insufficient erythropoiesis. And lastly, there are also several causes as to why there is anemia. It may be due to blood loss, impaired RBC production, or accelerated or increased RBC destruction. Let us also recall your RBCs, or simply your red blood cells. These are also known as your erythrocytes, and it takes approximately 18 to 21 days for the burst forming unit, or BFUE, to mature to become an RBC. The mature erythrocyte has a lifespan of 120 days in the circulation. Now, your RBCs are usually described as biconcave, discoid shape, and is enucleated with an average size of 6 to 8 micrometers. Let us also go over with a recall on the definition of osmosis. When we say osmosis, this is a process by which the molecules of a solvent pass from a solution of low concentration to a solution of high concentration through a semi-permeable membrane. Now, if a cell is in a hypertonic solution, the solution has a lower water concentration than the cell contents. Thus, water moves out of the cell until both solutions are in equal concentrations. When placed in a hypertonic solution, a red blood cell will lose water and undergo crenation or shrinking. On the other hand, if a cell is placed in a hypotonic solution, it will take in water across their membranes until both the external solution and the cell contents are in equal concentrations. A red blood cell will then swell or burst and undergo hemolysis when placed in a hypotonic solution. Now, animals and human cells tend to do best in a isotonic environment, wherein the flow of water in and out of the cell is occurring at equal rates. Let's now go over to our main experiment, which is the osmotic fragility test. When we say osmotic fragility test, or simply OFT, this determines the fragility of the RBCs when exposed to osmotic stress. This demonstrates increased RBC fragility in the blood specimens in which the RBCs have decreased surface area to volume ratios. In this experiment, a blood is added to a series of tubes with increasingly hypotonic sodium chloride solutions. Now, in the procedure, a standard volume, usually 5 milliliters of fresh whole blood, is mixed with sodium chloride solutions ranging in different concentrations. To be able to achieve these ranging concentrations, a solution of sodium chloride solution and distilled water are mixed in a set of 12 test tubes with corresponding labels. Now, the labels of the tubes corresponds to the amount of sodium chloride solution present in the tube. So in this case, for example, in tube 14, there would be 14 drops of sodium chloride solutions, while the amount of distilled water will now depend by subtracting the tube number to 25. So in this case, in tube 14, 25 minus 14, so you will be dropping 11 drops of distilled water in this tube. And then the list goes on. So for example, in tube 15, there will be 15 drops of sodium chloride, and then there will be 25 minus 15, 10 drops of distilled water, and so on and so forth until you'll be able to fill out all the test tubes needed for the procedure. Once solution is ready, place a drop of blood on each tube, which is done in a uniform way and are mixed gently afterwards. After a 30-minute incubation at room temperature, the tubes are then centrifuged and results are either observed through its supernatant fluid and measured spectrophotometrically at 540 nanometers or just examined visually on a bright light. Now we have two methods of evaluating OFT. As mentioned, we have your Sanford method and your Dassey's method. For Sanford method, this is the semi-quantitative method wherein hemolysis is visually observed under bright light or natural light, while the Dassey's method is the quantitative method where hemolysis is measured via spectrophotometer at 540 nanometers. So in our experiment, we will be doing the Sanford method. Now, for the interpretation of results, we have two hemolysis that we need to observe: the initial hemolysis and the complete hemolysis. When we say initial hemolysis, these are the tubes that has a faint pink supernatant fluid. Okay? While when we say complete hemolysis, these are the tubes that has a homogeneous red fluid with no sediment. For the normal test results in osmotic fragility test, initial hemolysis would be observed at 0.45% salt solution, while the complete hemolysis will be observed at 0.35 to 0.30% salt solution. Abnormal test results in OFT may be observed wherein the initial hemolysis may be early observed with more than 0.45% salt solution. Now, when cells have decreased surface area volume ratio, they have a limited capacity to expand. Thus, when placed in a hypotonic solution, they can only take a small amount of water, leading to immediate bursting. Now, red blood cells with smaller surface area, such as your spirocytes, will rupture more quickly. Thus, it has a increased osmotic fragility and lesser resistance to hemolysis. Examples of conditions associated with increased OFT are hereditary spirocytosis and some acquired hemolytic anemia. On the other hand, red blood cells with larger surface area, such as your target cells and sickle cells, these are capable of absorbing more fluid prior to rupturing. Thus, it has a decreased fragility and more resistance to hemolysis. Your target cells are also known as your podocytes, platycytes, and Mexican hat cells. These are observed in the condition thalassemia. And your sickle cells is also known as your meniscocytes, are observed usually or associated with sickle cell anemia. Thus, Results with decreased OFT are associated with the conditions thalassemia, sickle cell anemia, and iron deficiency anemia. Now, your osmotic fragility test is not a specific test for hereditary spherocytosis. It may only be used as a screening or presumptive test in determining certain conditions. And aside from as it is not a sensitive test, your OFT is also time consuming and may lead to several errors. 1. It requires a fresh heparinized blood specimen collected without trauma to avoid hemolysis. 2. Age or old blood may cause false positive results. Your fresh whole blood is preferred for this test because it contains live intact red blood cells. These RBCs can be fragile and susceptible to damage, especially if the sample is old or has been subjected to improper storage conditions. Third, incorrect concentration of sodium chloride in the tubes will affect the final pH of blood in the solution, thus may affect interpretation of results. There may be an error also with the unequal distribution of blood in the saline solution and may lead to false results due to different volume per tube. Thus, it is highly recommended that one person should only do the dropping of blood during the procedure for uniformity. And lastly, the use of oxalated or citrated blood is not suitable because of the additional salts present in it. Sodium citrate, as its anticoagulant, can alter the osmotic properties of the blood sample, potentially affecting the results of the osmotic fragility test. The presence of citrate ions could interfere with the ability to accurately assess the fragility of red blood cells in response to changes in osmotic pressure. That ends our discussion regarding OFT or osmotic fragility test. Thank you.